Hey guys, I don't know what happened because I'm supposed to have our institutional account and it is supposed to be pro account, so I don't know why this ended. However, we can go on right before we uh, have this problem again. Okay. Okay, so when we have a patient who has leukocytosis, in this case, I'm giving you an example in which this patient will actually um, would have over 17,000 leukocytes, okay? And in your lab report, you're going to get, for example, if in most laboratory reports, you get the percentage of the different types of leukocytes. For example, in neutrophil cells, in this case would be, for example, 82%. So what do we have to do next? We have to do some math, okay? In this case, we will have to multiply the number of leukocytes by the percentage of neutrophils and divide the result by 100. And we will have the numbers, the cell numbers of neutrophil cells, okay? This is called absolute neutrophil count. In Spanish, recuento absoluto de neutrófilos, okay? So we have this number, and how do we analyze this? ANC has also normal ranges that go between 2,000 and 6,500 cells, okay? So in this case, we have more than the double of the top normal range, which would be 6,500, right? The double. So when you get this kind of numbers, you always have to think of bacterial infections. Okay, this is really easy, really basic. What about the same patient? Well, a different patient, but with the same amount of leukocytes. However, in this one, we notice we have, if we have a percentage of 65 of lymphocytes, we do the same thing, okay? We multiply the number of cells of leukocytes by the percentage of the lymphocytes and divide it by 100, and we'll get the number of lymphocytes. This is also, as the previous one, this is the absolute lymphocyte count, or ALC. And how do we have to analyze this? Well, ALC also has normal ranges that go between 800 and 5,000. In this case, uh, it's twice it is more than the double, right, of 5,000, because we have 11,000 and even higher. So when you have this kind of patients, you would have to think of viral infections, right? Neoplasms, because in Spanish would be neoplasias, right? Neoplasms, and some bacterial infections. Can anybody tell me what kind of infections you might have to think about when you have lymphocytosis? Hello? Okay, you will have to think about intracellular bacteria, such as tuberculosis. Okay, so tuberculosis is related to lymphocytosis. We will go on to the blood chemistry part. Okay, here we have, you already know this kind of things. However, well, that's why I want you to focus on the pronunciation of these molecules. Okay, we have glycemia, creatinine, urea, blood urea nitrogen, uric acid, sodium, potassium, and bilirubin. You know the normal ranges. What would be important in the case if our patient had higher glycemia? Besides diabetes, right? Can anybody tell me? Give me an example of anything we would have to think about. High concentration of glucose besides diabetes. Okay, that's homework for you. Creatinine, maybe. It's easy. Danitza Burgos.
Yes.
Okay, what would be related to creatinine? Any kind of disease?
Some damage at the kidneys.
Okay, for example, could you give me one disease? The most common one.
Una insuficiencia renal crónica aguda.
Okay, and how could you say that in English?
Acute.
Yes, acute.
Renal. I'm not sure.
Okay, in English would be acute kidney injury. Because lately in the past, I think 3 years, we do not handle the term insuficiencia, okay? We actually are talking about lesión, okay? In Spanish would be lesión renal aguda, right? And that would be in English acute kidney injury, okay? That is what you might have read about acute, well, it's acute kidney injury, right? AKI, right? AKI. And we would read it like AKI. We have a classification, right? About acute kidney injury, we have the AKI classification. We call it AKI 1, AKI 2, right? So that is what this comes from, acute kidney injury, okay? Urea also and blood and nitrogen also are related to it. What about uric acid? Danita Zambrana, any kind of disease that might be related to uric acid? Danita Zambrana, are you here with us? Are you still here? Okay, let's go on with Mariana Sanginés.
Well, yes, I'm here, but I don't know.
You might have heard or read about in Spanish gota, right? So in English would be gout. Okay, gout is related to higher concentrations of uric acid. Okay, Paola, do you know what how would you call a higher concentration of sodium than normal? Higher than normal. In Spanish, maybe?
No, I don't know.
Okay, you might have heard or read about hypernatremia, right? Or hyponatremia, the opposite. So that is how we use this, okay? Hypernatremia and hyponatremia. Also, that is the same concept for potassium, would be hyperkalemia and hypokalemia, okay? Okay, what about bilirubin? Danitza Burgoa, do you know what concentration from what concentration of bilirubin um, can we find jaundice?
Some disease at the gallbladder. The levels of the bilirubin are increased.
Okay, great. What do you know what jaundice is? Jaundice, jaundice.
Yes, is ictericia.
Great. So when we are talking about bilirubin, we relate it to jaundice, right? I'm going to ask you in Spanish. ¿Desde qué concentración de bilirrubina esperarías encontrar ictericia? Hello.
I don't remember.
Okay, no, it's okay. Maybe Brisa, who just joined back. Do you know from what concentration of bilirubin would you find jaundice in a patient?
Jaundice, sorry, is when the bilirubin is high.
Okay, so yes, yes, it has to be high, right? However, there is a point from which higher levels of bilirubin can we can find jaundice. I mean, no en todas las personas que tienen hiperbilirrubinemia hay ictericia. ¿Desde qué concentración de hiperbilirrubinemia encontramos ictericia?
I think more than 4.
Actually, it is over 2.5. Okay, when a person has a bilirubin concentration higher than 2.5, we can see jaundice. Okay. Okay.
Okay.
Paula is asking here, can be found in people have diabetes. I don't understand your question, Paula. Could you repeat it so we can get rid of the doubt? Okay, in the meanwhile, we're going on, okay? So we're going on with some microbiological tests. You know this from the second year, right? Which is gram stain, okay? Tinción de gram, okay? So the name comes from a Danish bacteriologist who was Hans Christian Gram. It, as you know, checks for bacteria at the site of a suspected infection and classifies bacterial species into two large groups: gram-positive bacteria and gram-negative bacteria, right? As you can see on your right, the bluish bacteria are gram-positive, right? And the pink colored like, right, would be gram-negative bacteria. We also have microbiological tests such as blood culture. Culture has a lot of meanings, right? In this context, we have cultivo, right? In English would be culture. So we could have either bacterial or fungal cultures. And it takes a while for this for the bacteria to develop, especially fungus, right? It takes a lot of time to develop. Okay, Paula is asking, it can be found in people that have diabetes, I mean high bilirubin. Not really, it's not that related. Uh, Diabetes would be more related to glycemia and glycated hemoglobin that we're going to talk about later on, okay? High bilirubin, as Danitza told us, could be found in a lot of diseases, such as hepatitis, infectious hepatitis would be, another would be an obstruction at the biliary ducts, okay? Could increase. Also, hemolytic anemia could be the cause of increased bilirubin concentrations. There's a lot. However, diabetes is, I don't think it's related to bilirubin concentration. I mean, diabetes, right? It is not that related to high bilirubin concentrations, okay? Okay, so as we were saying, we could have bacterial or fungal culture. For a bacteria to develop depends on the bacteria, right? And the medium it is actually supposed to grow in, okay? It actually takes like 3 to 4 days, in some other cases 5 days. But fungal culture takes a lot more time. And also, we also have one specific culture for tuberculosis. Okay, it takes months to grow to have your report back to you. Okay, here in Bolivia, we have a lot of tuberculosis. So that's why we know it takes months to get your report back. So, as you can suppose, the development of bacteria in a petri dish, that is how you call this, petri dish, okay? Does anybody know what procalcitonin is or what this is used for? Procalcitonin. Does anybody know or heard or read about it? Okay, Brisa.
It's like
Okay.
sorry. It's
No.
like tumoral marcador. I don't know how to say that.
And what would you use this for? What do you think this would help you with?
Um, in cancer.
Okay, actually no. Procalcitonin is actually the most sensible molecule that is related to infection, okay? We always ask for C-reactive protein, right? Proteína C reactiva or PCR, right? We always do it. It's like sign, an inspecific sign of infection or inflammation, right? If there's any part of a person's body that has inflammation, this CPR is going to be elevated. However, it could be for inflammation or infection, but procalcitonin is actually very specific for infection. Okay, so this is like the most important molecule and the one that is going to identify if there is infection and not just inflammation and or infection, just like CPR. This is really specific for infection. If you have if you suspect of an infection but it is not that clear, you could ask for procalcitonin, and if it's high, it's sure that this patient has an infection in any part of the body. Okay, so procalcitonin is specific for determining infection. Okay, this is a really important molecule, so take it into account and read about it. This is quite interesting, especially for when you have sepsis, a patient with sepsis. This molecule is going to be really, really sky high, as some people say. Okay. Some other tests. BMP. Does anybody know what BMP is? Brisa.
Isn't that peptidos?
Okay. P stands for peptides. Okay. What about the BN?
Natriuréticos, I
Oh.
think.
Okay. And B?
No, I don't know.
Okay. It comes from brain. Okay. In Spanish would be cerebro, right? Brain natriuretic peptide. In Spanish, péptido cerebral natriurético, right? However, Brisa, you were going the right way. What do you think this molecule might help you for or with? Again, well, we have 10 minutes left. Okay.
Right now I can't remember.
Okay, that's okay. Does anybody remember? Okay, so I'm going to give you a clue. Okay, you can see this heart, right? So brain natriuretic peptide is called that way, I mean brain, because this molecule was first found in the brain. Okay, someone just wrote, yes, Paula, you're right. You got an extra point for that. Okay, thank you. You're right. So as we were saying, well, Paula just gave us the diagnosis, actually. It is, this molecule is used for heart failure in English, which in Spanish would be insuficiencia cardíaca, okay? So how does this work? I mean, as I was saying, this molecule was first found in the brain. That is why it has the B, brain, okay? However, later, some researchers found that this molecule was actually produced by the heart. And why? Because well, as you know, in heart failure, there is like, well, this heart cannot pump in a correct manner the blood, right? So every time it pumps and doesn't pump the right way, so there's some more blood inside of it because it's not pumping the right way. So this heart gets real filled with blood, and the muscular cells start stretching like a rubber band, right? You know what a rubber band is, right? Una liga de goma, right? So they start stretching like a rubber band, and that is the main stimulation for these cells to start producing this peptide. As you can tell about the name, this is a natriuretic peptide, so increases the volume of urine by eliminating natriuretic, right? Sodium. By eliminating more sodium, increases the volume of urine to get rid of that excess of blood. Okay, so that is what this is used for. In a patient with dyspnea, right? In Spanish, dyspnea, right? Actually, that is the correct pronunciation in Spanish, okay? For you to not make mistakes. It's not dyspnea, it's dyspnea, tachypnea, orthopnea, okay? Not dyspnea, okay? It's dyspnea. In a patient with dyspnea, okay, to differentiate cardiac dyspnea from a respiratory dyspnea, we take into account the brain natriuretic peptide. If this is high, then we have our dyspnea that is related to the heart. And if we have a patient with dyspnea and his BNP levels are normal, well, then it is a respiratory dyspnea, okay? Glycated hemoglobin. Notice the pronunciation, glycated hemoglobin. Can anybody tell me what this is?
Is the hemoglobina glucosilada?
Okay, what is this used for? I mean, how is this molecule formed? Does this require any enzyme to join the hemoglobin and the glucose?
What is an enzyme?
Enzima, right?
Oh, okay. No, I don't remember if the enzyme, but I know that it is used for the diabetes.
Diabetes, okay. Yes, it is okay, related to diabetes, right? However, this is actually a reaction that does not need an enzyme. This is a non-enzymatic reaction, okay? Hemoglobin and glucose bind to each other and they do not need any enzyme. That is its characteristic, okay? And as I think it was Brisa, right? Was telling us it is used for diabetes follow-up. It's not for, well, it could be for diagnosis of diabetes. However, it is most commonly used for a follow-up, para hacer el seguimiento, right? For follow-up. Urinalysis. This is for determining the compounds of urine, right? What can we use this for? We are running out of time. Danitza Burgoa, what would you use this urinalysis for?
To diagnose diabetes in a patient. It's one of the ADA es
Criteria?
uno de los, sí.
Yes, yes, you're great. Okay, so here in this with this test, we analyze the products in the urine, right? So we can use this for different types of diseases, right? As Danitza just told us, it could be useful for diabetes because when the concentration of glucose is higher than 180, right? This is in Spanish, umbral renal de glucosa, right? 180. So when it is higher than that, glucose starts to appear in the urine. Okay, great. What about
Sorry, I thought you were talking about the other one, the
okay,
glycated
like
hemoglobin.
okay, it is also used for
Yes.
this, right? And this urine test is also used one of its uses could be for determining hyperglycemia, right? Okay, what about urine sediment? Anybody? Okay, it could be used for, and was anybody going to tell me anything? I think I heard something. Okay, urine sediment is actually for, well, after we get the urinary test, we have this urine sediment. We use the same urine and we see it under the microscope to see if there is some cylinders. Does anybody know what the tam horseful protein is? Tam horseful protein? Okay, this is a protein that is produced at the loop of Henle, okay? And when there is, for example, red blood cells inside the lumen of the tubule, they get attached to this protein, which is the tam horsful protein, and then we have, as a result, we have red blood cell cylinders, okay? And any kind of cylinder is related to tam horsful protein. We also have polymerase chain reaction for determining any specific type of bacteria or any microbiological agent. And beta HCG is for determining pregnancy, right? Okay, guys, I don't know why, but we have less than 1 minute. And this is uh, where we're going to leave it. Let's call it a day. And